Greetings everyone, I am Nur Fatiha representing my team, and in this video, I will present our innovation project entitled Targeting Interleukin seed as an approach to overcome an acquired radioresistance to improve cancer treatment. Cancer, one of the deadliest diseases there is on the face of Earth. It is a disease in which cells change and grow uncontrolled. For decades, it has affected the lives of millions of people. Breast cancer in particular are prominent among women, with approximately 1 out of 8 women were affected. In 2020, there are more than 2 million women suffered from breast cancer worldwide. And in Malaysia, around 8,000 incidents of breast cancer were reported in the very same year. Modern managements include surgery, immunotherapy, chemotherapy, hormone therapy, and radiation therapy. Although results are promising in most cases, however, some were not responding to treatment. Recent studies reported series of breast cancer patients whom are not responding following radiotherapy treatment or the usage of ionization radiation to kill the cancerous cells. The culprit? Radioresistance. It turns out some of the cancerous cells develop immunity, instead eradicated upon exposure to the radiation. Previously, we innovated a model of radioresistant cell developed from EMT6 mouse mammary carcinoma cell line using fraction dose from gamma ray source, and these cells we term as EMT6RR-MGI. And currently, we are caving deeper into the underlying mechanism of why the cells developed radioresistance. Hence, our next innovation was inspired. Our objective is to investigate and determine the differential gene expression and pathways involved between acquired radioresistant and EMT6 parental cells. By isolating cells of two different attributes into two different groups, namely parental and EMT6RR-MGI cell, we isolated the RNA from both groups and we performed next-generation sequencing to further identify expressed genes pathway involved. This was followed by polymerase chain reaction or PCR to validate the result. Gene pathway analysis showed more than 20,000 genes were identified, in which 2,896 genes were expressed in EMT6RR-MGI cells, with KAGG pathway analysis showed that the pathway involved include T-cell signaling, chemokine signaling, IL-17 signaling, autophagy, and TNF signaling pathway. While 256 genes were expressed in parental EMT6, and pathway analysis showed the significant pathway involved are NF-kappa B, necroptosis, and apoptosis. Both groups equally expressed a total of 17,516 genes, with further analysis identified 418 genes significantly upregulated and 430 genes were significantly downregulated. And pathway analysis showed they were mainly in general oncogenic pathway. From the analysis, we shortlisted 16 genes followed by performing RT-PCR. The results showed 5 out of 16 genes were significantly expressed in the EMT6RR-MGI. Out of these 5 genes, IL-6 showed the highest fold changes, approximately 8 fold changes. IL-6 was upregulated in various chemotherapy-induced resistance in cancer cells such as gastric, breast, renal, and lung. However, there is a limited findings on radioresistant events were reported. In conclusion, we reported the upregulation of IL-6 gene in EMT6RR-MGI cell can be used to investigate cancer cell resistance. In the future, targeting IL-6 pathway may be beneficial in combating radioresistant incidents to improve cancer treatment and prevent cancer relapse, and give positive impact towards socio-economic and, more importantly, toward society. This study involved a collaboration with University Putra Malaysia or UPM and University Kebangsaan Malaysia or UKM. In addition, we have won gold in faculty level and silver during IIDEX 2020. With that, thank you.